Gentlemen, good afternoon. It's wonderful to see so many of you here this afternoon at the university, particularly in this fine weather. I'm Patrick Hackett. I'm the Deputy Vice-Chancellor here at the University of Liverpool, and I am delighted to be here tonight to welcome you to one of our open house events, Nanomedicine at Liverpool, Impacting World Global Health. Open House began last year and saw us opening our doors to hundreds of visitors keen to take part in a wide range of activities, showcasing the breadth and diversity of activity here at the university. This year, we are building on that success, and Open House has returned for a 5-day festival of talks, performances, exhibitions, and interactive experiences, showcasing great ideas, knowledge, and culture. We have already held a series of wonderful events this week, including hearing Ken Hom speaking at the World Museum Liverpool on Chinese values in a modern world, and an exploration of the late Stephen Hawking's work. And tomorrow, there is an opportunity to join in family fun as part of our contribution to the city's Light Night programme. This will see us reenacting the Battle of Actium, bringing mummification to life, and much more. But back to tonight. Nanotechnology is a fascinating area of science which has attracted significant global attention. And here at the University of Liverpool, two of our leading academics, pharmacologist and HIV expert Professor Andrew Owen and materials chemist Professor Steve Rannard, have formed the Liverpool Nanomedicine Partnership. Nanomedicine is the application of nanotechnology to the prevention and treatment of disease in the human body. Benefits have already been achieved across a number of clinically used therapies and diagnostics worldwide, with over 45 FDA-approved medicines reliant on nanotechnology for treating patients with conditions from mental health problems through to cancer. This evening, Professor Steve Rannard will explain how the Liverpool Nanomedicine Partnership has been working towards a range of novel therapeutic conditions over the last 9 years, with a focus on chronic dosing and infectious disease. He will also explore the scope of nanomedicine to further transform healthcare, with a particular focus on HIV nanomedicine for children. Steve is a leader in his field. He is the co-author of over 120 scientific publications, the co-inventor of over 90 patent families, and has attracted collaborative grant income of over £23 million to date. Steve has also co-founded the British Society for Nanomedicine and the Journal of Interdisciplinary Nanomedicine. So, without further ado, could I ask you in joining me in welcoming Professor Steve Rannard.
Okay, the problem with the build-up like that is I'm going to feel wholly inadequate. So firstly, thank you for coming. I've got more people here than I usually have turn up for an undergraduate lecture, so it could be that they know something more than you do about what you're about to see. Right, so we're going to talk about nanomedicine, but before we talk about nanomedicine, we need to talk about size, and we need to talk about and understand what nano actually means in terms of the number, as it were. So what I'm going to do now is just run through a few examples of things that have different sizes, things that you may be familiar with, some things that you won't, and I'll take you through the different length scales that lead into the nanoscale. So what I've got here is a length scale here, starting at 1 meter and going back down by a factor of 10 in each case. So we've got 1 meter, then into 100 centimeters, down into 1 centimeter, and down into a millimeter. And I'll put some scales on there just to help you sort of guide you through that. Is there any way we can drop the lights a little bit? Thank you. So let's start at the top. So we're in Liverpool, and football's a big thing in this city. So a standard football is about 25 centimeters in diameter. And because we're in Liverpool, I've got to put up both footballs. So there's a Liverpool Football Club and Everton, and apologies to the Everton guys for not doing too well at the moment. So that's about 25 centimeters across. What I've just put up now is a flea. sort of thing that you find on your cat or your dog. And they're about 3 millimeters across, so they're pretty small if you've ever seen one, and you know the sort of size that I'm talking about. What you're looking at now, this little picture here, is a scanning electron microscopy picture of a human hair. Okay, and a human hair is, again, a factor of 10 smaller than a flea, and is about 100 microns across. Okay, a micron being 1,000th of a millimeter. That's a red blood cell. Okay, so you've seen blood, but possibly not the blood cells themselves, and that's a red blood cell. It's about 7.5 microns across. Okay, so you could pack probably about 10 of them across the width of a human hair. That guy there, falsely colored, but he does look pretty, is the influenza virus. Okay, so that virus is about 150 nanometers in size. So a nanometer is 1,000th of a micron is 1,000th of a millimeter. And if we blow up this region here from 100 nanometers down, that's the HIV virus. Okay, a little bit smaller than the influenza virus, and we'll come back to him a little bit later. And what you're looking at here are titanium dioxide nanoparticles that are about 50 nanometers in size. And most of you will either have come in contact with them or deliberately applied them to your skin to prevent sunburn. Okay, so these are some of the things that are put into sunscreen and help protect you from the harmful rays of the sun. So let's go a little bit smaller. That's DNA. So what you're seeing here, this squiggly blue line here, this is an atomic force microscope picture of a DNA strand, and DNA is about 2 nanometers across, 1 nanometer being 1,000th of a micron. And those are xenon atoms. Okay, each one of these little white balls that you can see here is a xenon atom. They're about 130 picometers in size, and they've been arranged to spell the letters IBM. And clearly IBM did that. If we go a little bit smaller than the atom, what you're looking at there is a model of what comes out of a lead atom when you hit it, when you collide a high velocity proton into it. So these are all subatomic particles, and they're down in the 10 picometer and lower level. Okay, so there's an awful lot of space between the football and the subatomic particle. And what we're going to be talking about is anything less than a micron in the rest of the talk. The problem with talking about millions and billions and hundreds of millions is that we do it all the time in the news. The national debt is a big number, for example, and the hundreds of millions it costs to build something. But we kind of lose track of what that means. So if you look at 1 nanometer, that's a billionth of a meter. To put that in context, okay, if I take one step like that, that's about 80 centimeters. So 1 million steps is 528 miles. Okay, and to put that in context for you, that's like walking from Liverpool to Dortmund in Germany, including the English Channel. So you have to hold your breath from there to there, I guess. So that's 1 million steps, seems like a long way. So what's 1 billion steps? Okay, 1 billion steps is 528,000 miles. So if 1 nanometer is this one step I've just taken, a meter is 528,000 miles. Okay, that's the scale, that's the sort of length scales that we're talking about. So can I put 528,000 miles into perspective for you? It's about 2.1 times the distance to the moon. Okay, that's the difference. And to put the distance of the moon in context for you, you can fit the entire solar system into that distance. Okay, so it's a huge distance. 528,000 miles is from here to here and back again, and 0.1 added onto that. So distance is relatively easy to get into your mind, but if you like time instead, 60 seconds is a minute, 1 million seconds is 11.7 days. So what's 1 billion seconds? It's 31.7 years. Okay, so when I talk about nanoscale, I'm not just saying a little bit smaller than a meter, it's incredibly smaller than a meter. So that's distance, that's time. What about cash? So if we talk about lottery wins, 2016, some lucky person, a single person, in the UK won £66 million. In the same month, in the US, single win, somebody won $1.5 billion. Okay, imagine the difference. Seems like it's a lot of money anyway, but think about the difference between the million and the billion here. It's huge. To really depress you all, UK national debt was £2.02 trillion as of this time yesterday. Okay? And it goes up at £5,000 a second, which is quite scary. If you really want to watch that, if you want to depress yourself and watch that happening in real time, you can go online to watch the nationaldebtclock.co.uk. Anyway, so what is nanotechnology? Really, it's a tricky subject, nanotechnology, because it's not one thing. It's a combination of a number of things. Okay, this is Richard Feynman, who's widely acclaimed to be the first person who really started talking about working in the sort of kind of length scales that I've just been describing, really going down into the lower limits of where mass exists. So Richard Feynman gave a plenary lecture and wrote an essay in 1959 called "There's Plenty of Room at the Bottom." As a physicist trying to explain that there's plenty of innovation that can happen if you go small rather than going big. Norio Taniguchi in 1974 used the term nanotechnology for the first time. And what you're looking at here is a piece of Richard Feynman's plenary lecture that's been written by an atomic force microscope, and this was written in the I think it was between 2000 and 2010 in Chad Merkin's lab in the States. And this is actually a piece of his talk. The L there in the word people is only 60 nanometers across, and the D here is just 400 nanometers across. It's quite an amazing feat from 1959 to 2010 to 2015 to go from thinking about the nanoscale to being able to do things like this. The two guys down here, though, really started the experimental nanomedicine or nanotechnology happening. This guy's Harry Croto. I was very fortunate to have Harry teach me as an undergraduate at the University of Sussex. And this guy here is Richard Smalley. And the two guys collaborated together with other people, but these are the widely reported or regarded to be the leaders of the work. And they found for the first time a molecule. This is carbon-60. You might have heard of Buckminster fullerene. This is the molecule that they found. It's all carbon. It's the third allotrope of carbon. And you can see Harry here has built several versions of it on his desk. So just for balance, that's a red one, and there's the Everton one. But Richard, I think, has given the best description of what nanotechnology is. He says, nanotechnology is the art and science of building stuff that does stuff on the nanometer scale. And that's pretty much all you need to know about nanotechnology. Is it new? It's not new at all. So what we have here are stained glass windows from Augsburg Cathedral and York Minster. And the colors that you see in stained glass windows are not dyes, they're not pigments. They're nanoparticles of gold and silver. And the red here is silver, and the yellow here is gold. Even the blue over here is silver, and the different sizes give you the different colors. Okay, so all the way back at 1065, when they were building these beautiful windows, they were making nanoparticles to color them. And not only for color, the Damascus saber blades, if you've ever seen one, I don't know if you can see this beautiful pattern within the metal. That's driven by nanoparticles, nanorods, nanotubes within the actual steel, which make it extremely strong, even though it's very light and very, very thin. So people have been using nanotechnology without knowing it for an awfully long time. So what's nanomedicine? This is the best definition that I've seen of nanomedicine, and it's the medical application of nanotechnology, and I just told you nanotechnology is doing stuff at the nanoscale, but it's used for diagnosing, treating, preventing disease and traumatic injury, relieving pain, and or preserving and improving human health. Okay, and where that came from is me taking the best bits of all of the rubbish definitions that are out there And there are a lot of rubbish definitions. There's also an awful lot of rubbish in general about nano. So these are available if you really want to go looking for them on the internet. And this is literally rubbish, and I'm going to take a short period of the talk to show you why it's rubbish. But each one of these sites actually uses the term nanobots, okay? And you'll all have heard nanorobots, or at least the concept of them, and people have talked about them for years. And it's just fiction, okay? And I'll show you why. So here's some fiction. These are nanobots in the movies. So this movie over here is G.I. Joe, The Rise of Cobra, absolute classic. And this is a relatively recent one called Transcendence, which some of you may have seen. I've got to ask, anyone here from France? One. Okay, so I apologize for what I'm about to show you. So what you're seeing is a little clip from the movie where nanobots have been shot at the Eiffel Tower. Okay, so I'm very sorry about that. And I will stop this before the Eiffel Tower collapses. Okay, but the nanobots have been shot onto this. They're rampaging around Paris now, dropping onto anything and everything, eating it all up, and causing all sorts of havoc. Okay, so that's kind of a popular image of what nanotechnology is and what it could be in the future. So I think I'll stop it before it gets there. The internet is also full of these images. Okay, so this is what people often have in their head as being nanomedicine. These little machines running around the body doing different things. Okay, so these guys here apparently are cutting hair. Okay, I don't know why scissors are not good enough. These guys here are cleaning teeth. Okay, or at least considered to be possible to clean teeth. I dare say they don't stay on your teeth for very long, especially when you're in Starbucks or McDonald's, and then they go and clean something else, I guess. I don't know where they end up. But these other images are very, very popular of them treating disease, okay? And I'll show you why this is not possible. But this is Richard Smalley again. This is Eric Drexler. And Eric Drexler wrote a book called The Engines of Creation, The Coming Era of Nanotechnology, where he talked about self-replicating nanorobots in the world doing things. Eric and Richard had a huge debate, an argument for many, many years before Richard died. And Richard spent a lot of his time in schools in the US trying to convince children not to be afraid of technology. And these are actual excerpts from open discussions that they had in the literature. So here's Richard talking to Eric. Self-replicating mechanical nanobots are simply not possible in our world. You and the people around you have scared our children. And he was going into schools to try and prove to children that this was not possible. Let me show you some of the public perception of nanotechnology. This is Covent Garden. I've blanked out the name of the shop. If you zoom in on the shop window, you see this bench, and it says, "Health and beauty is happiness," which explains why I'm such a grumpy guy. But if you zoom in a little bit more into the window, look at this. No parabens, no nano. Yeah, that's a sales point for this little business here, that they don't sell anything with nano in it. Well, of course they do. They just don't know. Yeah. What they haven't got is nanobots, and I'll explain why. Okay, so this is one of the popular images in the literature and on the internet of a little nano robot sucking up cholesterol out of some gunked up artery somewhere. Okay, now, if I shrink down to being 50 nanometers in size, okay, and I'm inside this artery, you probably can't see it, but that little dot down there, that little red dot by my foot, is a water molecule. Okay, and water molecules are about 1/4 to 1/3 of a nanometer across. Okay, and they look like that, so a little bent molecule. What I tried to do was search the internet to try and explain what's not being shown in this image. So if I'm at 50 nanometers, there isn't just one water molecule. I'm completely and utterly surrounded by these water molecules. Okay? Now, luckily, this guy has done exactly what I needed. This is Mark Rober, who's an American engineer, and I understand that, but he's also a YouTube personality, which I don't understand at all. And what he's done is he has taken some things that you can buy called Orbeez. I didn't know these existed. These are Orbeez up here. There's the logo. Him and his mate here have got bags and bags and bags of Orbeez. Okay, and Orbeez are little swellable beads. And they've absolutely filled his swimming pool with them. Okay? And what they do is they swell, they absorb the water. And they put, must be millions of them that they've put into the swimming pool. And overnight they swelled, and now his swimming pool's full, okay, of just swollen beads. Now that's not that different to being surrounded by water molecules that you can now see because you're small enough to be able to see them. So let me show you what it's like in that environment, okay? So imagine that this guy is 50 nanometers in size. That's what... Turn that off. So what you're looking at is somebody of the order of magnitude of this guy surrounded by molecules of the order of magnitude of water. And as you can see, they can go exactly where they want to go all the time. They can choose to go from A to B whenever they can. Look how hard it is to move in this environment. That's what it's actually like if you're in the nanoscale surrounded by water molecules. And it's even worse than that, because water molecules move. Oh, wait. So water molecules are moving all the time, like that. So what it's actually like, if you're in the nanoscale, is not with all these static molecules around you. You are getting bombarded all the time by those water molecules. And if they're flowing from A to B, you're going to flow with them. Yeah? You're not going to be able to say, no, there's a disease cell, it's up back there somewhere. Yeah, you can't just decide to go and work on some disease that you found in a particular cell type. So this is utter fiction. It's never going to happen. One way to work out whether things like this can happen is to look at what nature's done. This is the smallest, well, it's the third smallest flying insect. It's called a fairy wasp. It doesn't fly very well. This is one of the best quotes that relate to the fairy wasp. They're not very strong flyers. They tend to be carried along on air currents. That's exactly the point. They can't choose to go over there. Okay? So rampaging nanobots chasing me, a little wind is going to blow them all away. Okay? How big is the fairy wasp? But this is a two-scale image. This is a fairy wasp. This is an amoeba. This is a protozoa. These are single-cell animals. And this is a multi-cell animal. It's quite an amazing feat of nature to be able to build that autonomous animal. Insect. This is the smallest animal. This is called a tardigrade. And there he is walking. See, somebody did it. There's always one person that goes, when that's on. He's beautiful, isn't he? So this is the smallest animal. When they're born with all the cells that they have. And the cells just get bigger, and that's how they grow. Okay, so again, what you've just seen is him walking about 20 microns. Okay, so rampaging nanobots coming at me. Oh dear, it'll be another week now before they get to me. Yeah, so not really going to be a concern. So, what is nanomedicine? Nanomedicine really isn't easy to define in a single thing. But you can break it down by administration route, so it could be orally dosed, inhaled, parenteral, so injected, nobody likes that, or topical or transdermal. You could break it down into two separate things, which is the vehicle itself. So what does the nano object look like? And it can either be a solid lump of poorly soluble drug, poorly water-soluble drug, so a nanoparticle made out of drug, or you could make a little entity, a little ball of something, and put the drug in it or bind it to it. Okay, so we're going to focus on these we're actually going to focus on solid drug nanoparticles for the rest of the talk. But you can characterize it in lots of other ways as well. So Patrick mentioned that there are a number of FDA-approved nanomedicines already being used in the clinic. Not a single one of them has nano written on them, okay? And the reason for that is twofold. One, people are worried about nanotechnology because people keep drawing pictures of nanobots, causing havoc. So they don't write nano on anything, and nanomedicine is just medicine when it's in the clinic, okay? It doesn't matter whether it's nano or not. So these here are all nanocarrier-based nanomedicine, so these are entities which have drug in them or on them, and these ones over here are solid lumps of drug in the nanoscale. Okay, so that's just a few of them. So what are we trying to do with nanomedicine? There are two main options. What we've got here is someone who's got a localized disease. In a particular place, you can point to where it is on the body, and what you'd like to do is dose that person with a drug which doesn't go everywhere. It accumulates in the disease site, and then that person is cured, okay? And the option, or the example here would be cancer, for example. If you've got a tumor in a particular place, you want a highly toxic drug to kill the tumor and not the healthy tissue. These sorts of nanomedicines are normally injected. You normally go to a hospital in order to have these injected. The alternative is that you've got a systemic disease, so that's everywhere. Okay, you can't really point to where it is because it's everywhere in the body. But what happens if people take the same drug orally is that they have different amounts. Every one of us here would have a different amount of drug that actually gets into the bloodstream from the same amount of drug in the tablet. Okay, you'll just metabolize it, absorb it differently. So this interpatient variation can lead to one person having a dose from the same tablet which might be a little bit toxic, and another person having a dose which has no therapeutic benefit at all. They just didn't absorb enough drug. And what we try and do in nanomedicine is optimize the amount of drug that gets into the bloodstream so they get the right dose and then they're healthy. Or at least they maintain potentially a chronic disease. That's normally orally dosed. As Patrick said, for some years now we've been working as a nanomedicine partnership here at Liverpool. It's a multidisciplinary cross-faculty team. And all I've got here is the lead academics, but we've got a big group of very dedicated researchers that work with us. And we have a collective goal, which is to do the best quality science we can, but really focusing on translation into actual clinical benefits, actual medicines. And our focus is infectious disease. Just a couple of slides on what we've been doing as a partnership over the years. We created the British Society for Nanomedicine in 2012. This is our website. We had 20,354 visits in the 16-17 year, and we had 70% of those visits were new visitors from all over the world. Got about 750 members now, and we have an annual early career researcher meeting, which was in Belfast last year, and it'll be in London this year at the Crick Institute. And we've launched an international journal. We've also done a number of scientific things, obviously, and since 2008 we've been funded very generously from various organizations, funding organizations. We've generated collaborative interactions with a bunch of companies. We're in a global consortium which is focused on HIV. We've got some international and national collaborations, and we're the sole UK representative in the European Nanocharacterization Laboratory here at Liverpool. So what about the science? Let me show you some science that we do. I'm going to show you two short pieces of the science, two short summaries of the science that we've been doing. One on HIV oral dose optimization, so that's trying to optimize how much drug goes into the actual medicine, into the pill. I'm going to show you a little bit on malaria prevention. And I know I've seen a couple of the guys from the group in the audience, and it's a huge apology for not highlighting the work that they're doing as well, which is different to what I'm about to show you. So, HIV. What I've done here is I've screen captured what I think is some of the most irresponsible reporting I've seen on UK national television for an awfully long time. And I only know it's irresponsible because I'm working in the field. But last year, the BBC ran a story, which is HIV life expectancy near normal thanks to new drugs. And I watched it over my breakfast on BBC Breakfast News, where they brought in some HIV patients, people who are infected with HIV, and they basically said, oh, isn't life wonderful? You don't die of HIV anymore. Okay? Well, this is where I grew up. That dates me completely. But these are tombstones on TV with AIDS written on and posters saying my boyfriend gave me AIDS. I was only worried about getting pregnant. At that point, it was a life sentence. It's not anymore. In the Western world, let's give you some reality on HIV. So in 2015 data, and it takes a long time to generate these data, so 2015 statistics, there was an estimated 36.7 million people globally living with HIV, and the estimate actually ranges up to 40 million. There's no cure for HIV. Once you're infected, there is no cure. In that 36 to 40 million, there's 3.2 million children, and children are defined by anyone under the age of 15. In that year alone, there were 2.1 million new infections globally, okay, for a disease that has no cure. The vast majority of the burden is not the rich West, okay, it's the developing nations. 25.5 million of this 36 to 40 million live in sub-Saharan Africa. 1.1 million people died in 2015 alone of HIV and AIDS-related diseases. Okay, so that's a huge number of people. So to have a report on BBC News saying it's okay, everything's fixed, it isn't. It really isn't. One thing I'll say before I move on, 2.1 million new infections, 1.1 million people died. Okay, that's 1 million people who survived. Okay, and are living with HIV, 1 million new people in that year. Just as a comparison, everybody knows about the Ebola outbreak, and it's in the news again now, and everyone's petrified of Ebola, and they should be. But in 2014, the CDC, the Center for Disease Control, confirmed a total of 11,000 deaths in that entire period when the Ebola outbreak happened. Okay, 11,000. HIV victims in 2015, there were 21,000 a week. Okay, not over the entire period of the Ebola crisis. 200,000 children per year die of HIV. Okay, and in that year alone, 40,000 new infections and about 250,000 children newly infected in that year. Okay, so the Ebola outbreak was terrible. It was globally discussed, and HIV you barely see in the news anymore. Total numbers so far, estimated 78 million people have been infected, and about 35 million people have already died. And it's worse than that. So this is the problem that the healthcare systems are dealing with. I just showed you more people got infected than died of HIV. So there's a huge and increasing cost on healthcare systems to dose therapies to those people with HIV. And the drug supply is barely matching the demand. The great success of HIV therapy is that people don't die, or not as many people die. That means more people are living with HIV and need therapy to stay alive. A large number of those new infections are caused due to mother-to-child transmission. So this is HIV-positive women who are transmitting the disease to their babies. And there's not a lot you can do in some of the situations, the environments that the people are in. About 40% of people living with HIV globally don't know they have the disease, and only about half the people who do know have access to therapy. Okay? So the other problem is a lot of the HIV drugs that are key to therapy are poorly water soluble, so the doses are quite high in order to get enough of the drug into the bloodstream. And you can see here, this lady has to take this huge pill here, plus that huge pill, every single day. And that can be a real burden, and people don't like it. They stop taking their drugs, and that can lead to resistance. So for the last few years, we've been working here at Liverpool on a technology, what we call the solid drug nanoparticle technology, which takes poorly soluble compounds and turns them into nanoparticles. What we do is we dissolve them in a solvent that doesn't mix with water, so we've got an emulsion. We freeze dry the emulsion, take out the water and the solvent. We end up with a porous material which has particles of the poorly soluble drug in, and when we add them back to water, the matrix dissolves and releases nanoparticles of the drug. And the best way to think of this material here is like a blueberry muffin. Okay? So the blueberries here are the drug, and the muffin, the good bit, is water-soluble materials, excipients, we call them polymers and surfactants, which just keep them separate from each other. To give you an image of that, there's the blueberry muffin at the top, and when we add it to water, you can see what happens. This is a poorly soluble dye added to water, and it doesn't do anything. Okay? Same poorly soluble dye, but in this blueberry muffin formulation here, added to water, and what we get now is something that looks like a solution. And it's not a solution. That's particles floating on the top of water. These are particles as well, but they're just dispersed within the water. And what happened in the water is a bit like this. So what we have is the cake mixture, as it were, dissolving into the water and the blueberries being released. Okay? So that can happen in the stomach as well as in this conical flask. So why would you make very, very tiny particles of drug? Well, people have been doing it for years to try and overcome slow absorption of drugs into the bloodstream. So if you look at a 1 centimeter tablet, if you pack that 1 centimeter tablet with 100 micron, about the width of a human hair, lumps of drug, then they look a bit like this, about the same size as the features on a vinyl record. If you break that up into 10 micron particles, for every 100 micron particle, you get 1,000 of these. And they look like this. So the scale bar here goes from 500 microns down now to 50 microns. That's about the feature size on a CD. And if you break this 100 micron particle into 200 nanometer particles, that's about the feature size on a DVD, and you get 125 million of these for every one of them. Okay? So when it gets into the gut, into the intestine, it's blown apart into lots and lots of little pieces, and they can then be absorbed a lot easier into the bloodstream. And there's a close-up image of a milled product into the nano range. That's 1 micron. That's 100th of the width of a human hair, and hopefully you can see all the tiny particles that are in there. We've got a process which I showed you just now, the freeze-drying process, and we take drugs and polymers and surfactants, and we basically make as many different formulations as we can, because there are so many different options that you can make. So we screen. This is what it could look like physically. This is one of the screens that we put. There's 160 different formulation options there, all made in this machine, a freeze dryer. We produce the emulsions, we freeze them, we test them physically, then we give them to our pharmacology colleagues, and they tell us which ones are any good. Once we've gone around that loop a few times, the optimum ones are converted to a process called spray drying, and the reason we do that is because it's scalable. We go from our benchtop spray dryers through to semi-pilot plant scale, and then the powders out of here can be filled into capsules and dosed so that we can test their efficacy. And if we find one that really, really works, it can go onto mass production and out to the clinic in tablets. It's a bit like looking for a needle in a haystack, okay? Because there are so many different formulations that you could make. The question is, which ones are the good ones? So you could, in that situation, just go in head first, but we feel that we're a bit more technically able than that, and we try to narrow down as fast as we can into the formulations that work. These are actual nano formulations that we've made that were put into capsules and dosed to humans. This is Marco Giardiello, who took the process all the way through from that screening all the way through to this semi-pilot scale into capsules, and then his materials were dosed to humans a couple of years ago. And what we did here was make the world's first nanomedicine to try and help HIV patients. When we looked at the stuff in the lab, what we expected, what we modeled, was a situation where we were getting far too much drug into the bloodstream from the same dose. So these orange curves are what happens if you dose a particular drug to humans globally, entire population. You take the tablet, a bit of drug gets into your bloodstream, a bit more does, and then over time it goes away. 24 hours, you take your next dose, the drug gets into the bloodstream and it goes away again. That's why you have to take another dose every day. And what we saw here was from our nano formulation at the same dosing level, we were getting much more into the bloodstream. So what that suggests is that you can decrease the amount of drug and bring these curves down onto these orange ones. That's exactly what we saw in human trials. So efavirenz is one of the drugs we worked on. That's a 600 mg daily dose. That's the red line. The blue line is efavirenz, nanoefavirenz, dosed at 300 mg daily. And as long as it stays within this red line, it doesn't go below this point, then we're what's called bioequivalent. Okay, so what that means is we can half the amount of drug in any tablet. Do we save money that way? That's not what we're trying to do. What we're trying to do is take the same amount of drug, make twice as many tablets, and dose twice as many people. Okay? So it's not about cost saving. If you look at what happens if you go from 600 mg to 300 mg, you could save $471 million over 5 years, or you could free up production of 2,759 tons of drug in that same period of time, or you could use that same amount of drug to treat twice as many people. And don't forget, only about 40 to 45% of people have access to therapy right now. So those are the sort of savings for efavirenz and lopinavir. We're working now on darunavir and ritonavir, and the savings there are huge. It's a very expensive drug to make, and we're looking at potential savings of $300 million a year, per year, not over 5 years. So again, if we can half the dose of that, then we'll be onto a winner for global charities. I'm going to very quickly run through malaria. If you think HIV is bad, malaria, 212 million new cases of malaria in 2015. 212 million. Okay, it's a curable disease though. 90% of the cases were in the African region down here. A child every 2 minutes dies of malaria, which is quite a scary thing. Child less than 5 years old. And every year, millions of people travel from these green areas to the red areas and hope that by taking a tablet, if they get bitten by a malarial infected mosquito, they won't contract malaria. What we've done is working with Johns Hopkins, the #2 medical school in the world in the States, is we've worked on a drug called atovaquone. And atovaquone won't cure you of malaria, but it is a good way to prevent malarial infection happening if you get bitten. Okay? The problem is if you take your tablets correctly, you build up a resistance or an amount of drug, a concentration of drug in the bloodstream, which will keep you in the green zone. That will keep you protected from being bitten. If you forget to take a tablet, maybe take it a bit late, take it a bit early each day, you may well just dip into this red zone. And if you do that, then the malarial parasite will potentially take over and then you're infected. So this is how malaria basically works. I'm not an expert in malaria, but this is the mosquito. He bites somebody, they're infected. The protozoa end up transported to the liver where they turn into the parasite. The parasite then blooms in the blood and you get the classic symptoms of malaria. An uninfected mosquito will bite this person, and then he'll bite this person that doesn't have malaria, and that's how it gets transmitted. So there's only really these two vectors you need to really consider. It's humans and mosquitoes. If you take your tablets, this could happen. But if we could give you one injection, and that slowly doses anti-malarial drugs into your bloodstream, then you could be protected potentially for weeks. Okay? And it's not then reliant on you being very adherent to taking that therapy every day. So we made a bunch of nanoparticle formulations. Atovaquone is very poorly soluble. We dispersed it into water, the blueberry muffin that I showed you a minute ago, and we tested from all of these formulations different drug release rates. We picked a few. We looked at these releases in this way, so this is varying the amount of drug in each of these formulations. And then we tested it in a mouse model where we injected the mouse with solid drug nanoparticles into the muscle. Okay? And this is what happened. We saw drug in the bloodstream all the way out to 35 days after the injection. Okay? So not just tablets every day, one injection and drug in the bloodstream for 35 days. So does that protect them from malaria? Well, at 200 mg per kilo dose, all of the mice that we injected were malaria-free if you bit them with a malarial mosquito on day 28 after the dose. Okay? So at least for a month, they were utterly protected from being bitten by malaria-infected mosquitoes. So that's cool. Even more cool is the drug itself is metabolized by mice 5 to 7 times faster than humans. So if I gave you the same injection, you might be protected for 6 months. Okay? It's a huge benefit. We're trying to get people excited enough to fund us to do that human trial. So all the way back to HIV very quickly. What about the children? I told you all about the children with HIV. What about therapies for them? Well, this here is Colectra solution. You can't give tablets to children, especially when they're very, very young. So this is Colectra solution, which is poured onto a spoon and fed to children every day from the age of about 3 weeks old until they're about 8. Okay? So what does that formulation look like? Because I told you that the drug is poorly soluble in water. Well, that's a mixture of ethanol, 42.4% ethanol, 15.3% propylene glycol, that's to dissolve the drug, and it has limited stability above 25 degrees. And where do we need it most? Sub-Saharan Africa. Yeah? So that's like taking a poorly soluble drug and dissolving it in a mixture of gin and antifreeze. And that's it. That's what they've got. That's the formulation. So the nanomedicines that we have disperse into water. We don't rely on solubility. We make particle dispersions in the nanoscale. So we could, with the programs that we're working on here, revolutionize pediatric HIV therapy in the developing world. The problem is that globally there's almost no funding for pediatric HIV therapy development. The reason for that? Well, you've seen the growing numbers of adults that have the disease. Okay, so those are the big numbers, and that's what the big charities and the big companies are focused on. So most of the children that become infected die before they're 5 years old. Okay, the other ones are being given a cocktail of gin and antifreeze. So we've been very lucky. Christina and her team at the front here have been helping us fundraise to try and do a pediatric trial of the things which we've already been paid to develop. And already we're up to about £170,000 of alumni donations to this, which we're incredibly grateful for. We need about 400,000 to get to the point where we might be able to actually run a trial with the actual formulations which we've already developed. So I need to finish, otherwise Patrick won't have any time to run around to the next one of these that he's going to. But the nanomedicine team has been huge over the years. This is some of the names from the group. That's the partnership itself, and these are some of the people that have collaborated with us and funded some of the work. I wanted to finish on one lighter note, though, because I know I've kind of depressed you all at the end. This is what I believe to be the first description of nanotechnology in the actual published literature. Okay, this guy here, eminent scientist, is Dr. Zeus, who wrote the book, The Cat in the Hat Comes Back. And if any of you have read it, you know exactly where I'm about to go. Okay, well, I hope so. So very quickly, quick synopsis of the book. This is the Cat in the Hat. He breaks into someone's house. He has a bath. I don't know why. But he leaves a stain in the bath. Okay? And he's pretty sad about this. So he decides to clean the stain off the bath. and he uses a nightdress that he finds in one of the bedrooms, and he converts the stain from the bath into the stain on the nightdress. He's not very happy about that. So how does he get it off the nightdress? Well, he realizes he needs help. So he takes his hat off, and under his hat is little cat A, and under his hat is little cat B. And A and B and C actually help him move the stain from the nightdress onto the bed, and then the bed onto the television, and then eventually they throw the stain out the door. Okay? Well, that causes an environmental issue. So they need little cat E, F, G, H, I, J, K. You can probably see where it's going. They go outside and try and clean up the local environment, but it gets worse. This is a global disaster now. So they keep on taking hats off. Eventually, they go all the way down until finally there's little cat Z. Z is, I guess, the right way to say that for the book, but Z is what I'm going to use. So they're looking at little cat Z. What on earth can he do to help? Well, what's he got under his hat? He's got Voom under his hat, okay? And what does Voom do? Fixes the problem, okay? Bingo, everything fixed. So when my two children were young, and they'd hate to know that I'm putting that picture up right now, they did ask, how big is little cat Z? Well, if you assume that the cat in the hat is 2 meters tall, and they half in size as they take their hat off, little cat Z is 30 nanometers in size. So that there is nanotechnology. Thank you very much. So I think we've got time for a few questions if anybody's got any. And there are some roving mics around. 2 roving mics, 1 at each end of the room. Anybody got any questions? That's one in the middle.
Oh, thank you. Thank you very much, Steve. That was a fascinating talk there. So I'm particularly interested in the HIV side of things. So nanomedicine in terms of the formulations that you make. One question is, how does that affect the efficacy of these antiretrovirals? And the second question is there a barrier towards drug companies developing this on a larger scale, especially, like you say, for the adults? You mentioned briefly Vavrins and also the PIs as well. Are you going to be moving towards the integrase inhibitors?
Yeah, good question. So the one on the right is a protease inhibitor. We are going to be working towards integrase inhibitors as well. It's interesting, quite a lot of the molecules that are being developed have got pretty good water solubility. And what we're focusing on is the ones which are really poorly soluble, because those are the ones that have to be dosed highly. What you're looking at here from an efficacy perspective is the amount of drug that gets into the blood. Okay, so you take the tablet here, the drug levels in the blood increase, and then over the course of 24 hours, they decrease back to where they started. Okay, that's the conventional dose. If that amount of drug in the bloodstream is efficacious, then just staying above here will do the job. Now, interestingly, where we are is a little bit higher at the end. So what that might mean here is that we might have a little bit more flexibility if somebody doesn't quite take the tablet 24 hours later. Okay, so if you imagine, if you put an imaginary line here, below which there's no efficacy, and you put another imaginary line there, above which you might be in a toxic area for the drug, keeping between those 2 lines, which is the therapeutic window, is exactly what we're trying to do. So we don't have to match this exact line, we just need to stay in the window for the 24 hours. Okay, if we can do that with half the amount of drug, then we're laughing. And what we saw was the same effect twice. So this red line here is the nanolipinavir, so another protease inhibitor. And here we're above the line, but we're not above the maximum value. And again, this is 42 hours. So at some point around here is where this would be deemed to not be efficacious. And we've got that little area where it may give somebody a little bit of flexibility to not have to take the tablet exactly on time. So as long as we're within the window, we assume that we'll have the same efficacy. I think your next point was how are we going to move this on? Well, if I can, let me try this. Yeah, okay. So in the consortium that we're in globally, what we have here is a group called the Medicines Patent Pool. And the Medicines Patent Pool are a UNITAID-funded, not-for-profit organization. And what they do is they gather together intellectual property, so patents protecting drugs, and intellectual property that protects drug delivery systems like ours. They pool it together, and they make it charitably available for the developing world. And what we've done is we've got our entire patent portfolio, and I think there are about 15 patents protecting our technology, and we've just put it all in the pool, okay? So what MPP are doing for us is they're bringing in suppliers of drugs which have patented drugs, and collectively we're looking for generics companies that will be licensing that for free and then making the potential therapies available to the developing world. So we tried to take the barrier to development and make it as low as possible. Does that answer? Yeah, cool. Yeah. We've got a mic coming at speed.
You were saying that you've got £23 million research grant.
Yes. So
how much of that is from Farmco? Pharmaceutical companies?
Oh, very, very little.
Yeah, because I can see no benefit for them.
No, there is. There's no benefit from a charitable perspective, which is what we're trying to push. But there is on drugs that they're working on. So there are some drugs, for example, by these guys up here, V, for example, which are right at the early stages of being developed. Now, if they can optimize the therapy first, then the tablets are much smaller and they'll be more easier to take as a therapy. But also, they won't be throwing away a load of drug. They could be treating more people. So the HIV world is quite interesting. It's highly dominated by very, very active patient groups, very vocal patient groups, who have, one way or another, forced the pharmaceutical world into a place where they are almost ashamed to make a profit. Okay? So the guys like Vive here, and guys like Cipla and Mylan are doing their utmost to work with the Medicines Patent Pool, UNITAID, Bill and Melinda Gates. I think the Clinton Health Access Initiative are on here too, to just try and deal with the problem. And the reason they're doing that is A, they don't want to be embarrassed. B, there is a global problem which they've had to wake up to, and it's not a profit-making opportunity for a lot of these companies. There's a company, Janssen, for example, who have donated drug to us. The arm of Janssen that works on HIV is loss-making for Janssen. So a lot of the companies have got on board with that. It's not the same for all drugs and all disease areas, for sure.
No, that's right.
Yeah, no, absolutely. But HIV is a very, very special case. Oh, there's a question at the back. There's a mic just coming. There we go.
Good evening. Thank you very much for the lecture. I was just wondering, if you're creating a drug that is going to have the strength,
Yeah.
what is the likely toxicity likely to be in the red blood cells of the patients, please?
That's again a very good question. And if I go back to the graph that I showed a minute ago, if we're still within this therapeutic window, then the same amount of drug got into the bloodstream, so the toxicity profile should be the same. Okay? So they shouldn't change. However, the drug that doesn't get absorbed by the conventional formulation does just pass through the GI tract, where it might have some localized toxicity in the actual gut itself. If we've half the amount of drug that's in the tablet, then it won't be half, but there'll be a reduced amount in the GI tract that's just passing through anyway and is poorly absorbed. So hopefully, we don't know this, but hopefully this would have a reduced toxicity profile in the actual gut itself, But in the bloodstream, it should be pretty much the same because we're still within that window. If this has any issues, this actual profile, then you never know, that might be a little bit better. But theoretically, there should be no real change on actual blood toxicity. Okay. Any other questions? Another.
Making a system that is more effective, or at least can be more widespread, is excellent. But I visited a number of those countries. Mm-hmm. Who is working on the distribution? Because there's a huge amount of ignorance, and the drugs may be there, but there's a great ignorance barrier to delivering to them, to the people who need them. Is there anyone working with you, or do you know of anyone who's doing that?
Yeah, so one of the projects which we're working on is funded by USAID. So this is the Agency for International Development. Okay, so that's in essence, well, it's funded by a group called PEPFAR, the President's Emergency Plan for AIDS Relief. Okay, and we're in a £50 million global consortium. And we don't get 50 million, it's spread around everyone. But these guys here, iBase and ICAP, these guys are patient advocacy and patient engagement groups. They're in the field teaching people how to take the drugs, because they don't always know how to take them, and they don't always know or understand the implications of missing a dose. So if you miss a dose with an HIV drug, you might get away with it. If you do it 2 or 3 times, you may well have dipped into that non-efficacious level, so that actually the therapy you were taking is no longer any good to you because you developed resistance to that therapy. Okay, so there's first line therapy, second line and third line. If you develop resistance to first line, you go on to second line, and that has worse side effects, so you know you're taking second line. If you fail on second line, you go on to third line. Those are the oldest drugs, which are not the best. If you fail on third line, there are no other drug regimens. Okay, and you've got to stick to or adhere to your therapy for your entire life, because you're not going to get cured. So what these guys are in the field trying to do is educate people, explain to them how it's done, explain the importance of taking their therapy correctly, and explain prevention of HIV transmission. The guys here, Mylan and Ciplar, for example, so in our consortium we have Mylan as one of the companies. They're not funded by the consortium, but they're part of it. And just to do a trial at the national level, so we're doing a phase 3 clinical trial, might take 30 million tablets to be produced. Yeah, because you need to do it for months, and you need a big enough cohort of patients. So the amount of effort that these guys put in is huge. And as I said, not all of it, well, almost none of it is profit-bearing. But you're entirely right. Education is a key thing. On that note, though, I'm about to get on a soapbox. HIV in Europe is not a huge issue. It's a problem. It's not good. But the UK has more HIV positive people than almost any other country, or we've had more infections than almost any other country since 2007. I looked at the stats the other day. We're about 9th on the list of 31 countries for new infections every year. Okay? So the education, even within the UK, of the risks of HIV is pretty poor, from my opinion. And the sort of scope and attention that things like Ebola get, when we're not even talking about HIV, or even worse, saying it's a problem you don't need to worry about, it just shocks me horrendously. The BBC portrayed it as a disease you could live with for the rest of your life. As long as you take tablets, it's fine. And I was horror struck. So education in Europe isn't great. Did that answer your question? Is that? Yeah? Okay.
There's still a long way to go.
Oh, there is. Huge. Absolutely huge. Have we got time for one more question?
Yeah, one more.
If there is one. Oh, there we go. So can we... So the mic's right next to you, so that's helpful.
You said that the children's formulation had to be stored at under 25 degrees.
Yeah.
Is that the same for your nano version?
No. So the nice thing about our nanoparticles is they're powders or granules. We're starting to make granules out of them now. So they're in the solid state, and you disperse them into liquids when you need them. It's actually keeping the liquid stable that's the problem. And that formulation is a disaster in many different ways, not least of which the contents. But the fact that it hasn't got any extended stability means they're throwing away drug that goes out of date that isn't used. And that's just wasting, good HIV drugs. So it's a problem that needs fixing. It's a problem that's widely recognized within the pediatric community and the HIV world. But when the numbers of adults are rising by 1 million a year, that's where people are focusing the attention. And it's potentially something that we've got an answer to. But no, we've got actually accelerated storage data for our own formulations. And we had to generate this in order to do our first human trial for 12 months of 40 degrees, 75% humidity for one of our formulations. And we've just, I think, so Alison's right there. We've just got to 4 months, haven't we, Alison, with our new formulations? So 4 months at 40 degrees rather than limited at 25. Okay.
Okay. Steve, thank you so much for such an inspiring session. I've got young twin boys, and they like to ask me, what did I do today, or what did I learn today? And my head was beginning to hurt, thinking, how am I going to explain nanomedicine to them? Well, thankfully, they love Cat in the Hat. So. But seriously, this is such an important topic. As you've heard, we here with Steve and his colleagues at the university are on the cusp of changing pediatric AIDS therapy around the world. We have the potential to offer a better, cheaper, life-saving treatment. As you've heard already from Steve, until recently there was no legal requirement for drug companies to produce specific products for children. And as the lifetime of patents that initially protected the drug development companies has now ended across a whole range of critical antiretroviral drugs, there is also very little commercial incentive for the progression of new child-specific HIV therapies. Therefore, the job of developing pediatric HIV therapy falls to academics like Steve and his colleagues, to charities and to not-for-profit organisations. And so this is why this project is one of the university's current fundraising priorities. If you would like to find out more, please speak to one of my colleagues for further information. But now, I'd like to close by thanking all of you very much for coming this evening. I'm pleased that you could join us, and I hope that you enjoyed the session as much as I have. You now have the opportunity to join us for a drink in the reception and to discuss this evening's event, and I'm sure that Steve would be happy to answer any further questions that you might have. I also hope that we might welcome you back to our campus soon to attend one of the very many public events that we run right throughout the year. If you'd like more information, you can visit our web pages and you'll see all the listings for future events. So I'm not going to keep you any longer. This concludes this evening's event. Steve, thank you so much. Please join me in thanking Steve for his